Hello, everyone, and welcome to another episode of Dr. Gilbert Hosts. Today, we're going to be meeting 3 APDA-funded Parkinson's disease researchers, and we're going to hear about their fascinating research, how it is bringing us closer to a fuller understanding of Parkinson's, and potentially how this research is involved in the development of new treatments for Parkinson's. And we're also going to hear about how APDA funding made a difference in this research in establishing these Parkinson's disease researchers' careers and how that funding continues to be important today. We're going to hear those behind-the-scenes stories of the critical importance of APDA research funding. So our 3 guests are Dr. Clemens Scherzer, Dr. Sunil Kumar, and Dr. Giulieta Riboldi. Each will tell us a little bit about their research, their career trajectory, and the role that APDA played. And I'm going to be introducing each one. Each one will give their brief presentation, and then all 3 guests will return and we'll have a Q&A. So get your questions ready for that. So our first presenter is Dr. Clemens Scherzer. He is the Stephen and Denise Adams Professor of Neurology, Professor of Genetics and Neuroscience, and all at Yale School of Medicine. He's the founding director of the Stephen and Denise Adams Center for Parkinson's Disease Research of Yale School of Medicine, which is an interdepartmental center which is building the future of precision medicine for Parkinson's through genomics and AI. He's also the director of the APDA Center for Advanced Research at Yale School of Medicine. In his clinical practice, he's the academic chief of the Division of Movement Disorders and directs the Yale-Harvard biomarkers study. Prior to joining Yale, Dr. Scherzer was the professor of neurology at Harvard Medical School. And at the start of his illustrious career, from 2004 to 2006, Dr. Scherzer was the recipient of APDA's George C. Kotzias Memorial Fellowship, which set him on his current career path. And he's continued to work closely with APDA through those years as a member of our scientific advisory board. Dr. Scherzer, the floor is yours.
Here and to get a chance to talk to the APDA community. Thank you, Rebecca, for having me. It's really a pleasure. So let's see if my slides work. Yeah. So we are, Chris, I think we're having an issue here with our slides. They're not showing what I'm seeing. Maybe let's restart this. Present, share screen.
So,
window.
Hi, everyone. So are we able to get the slides up, Chris, do you think, or should we?
I think we're good now, right?
Yeah. We're good now? Okay, fantastic.
Okay, this is a live show, so I think this makes it even more exciting. So we have a very ambitious goal at the Yale Adams Center and the Yale APDA Center, and that is to develop a precise predictive and preventive medicine for Parkinson's disease. And to do that, our dream really for the future is to have a patient be able to come to the clinic, take a few drops of blood, sequence the patient's whole genome, use all the electronic information in the electronic health records, and based on this big data, be able to predict what the glitches are in the patient's brain cells and what the future holds for this patient, and to recommend tailored medicines that turn all fast progressors into slow progressors and prevent bad complications from ever occurring. And the goal here is actually, I'm showing you here two patients who actually really was seeing at Mass General. One, after 15 years of PD, was able to play 165 rounds of golf every year. And the other one, with the same disease duration, was already in the wheelchair. So our goal is to predict complications, predict progression, and turn everybody into a patient who can play 165 rounds of golf 15 years into the disease. And so how are we going about this? Well, we have a very ambitious goal. We are decoding the genetic software of brain cells in Parkinson's disease and attempting to simulate them on a high performance compute cluster, and to use this to identify disease targets, design precision medicines, and prevent disease progression. And here, I'm just showing you a few snapshots of where we are with this. We identified with single cell genomics 92 cell types in brain regions important for Parkinson's disease. And we can map these cell types to their precise location in the brain, and to start making an atlas of the Parkinson's brain. And already some interesting results came out of this. For example, we discovered that although pretty much everything that's known about the human brain is based on just a function of 2% of the human genome, turns out that actually more than 64% of the human genome is active and transcribed and part of the software of brain cells. So only 2% have been studied, but actually 64% are active. So that's really important, and a lot of work to do to understand that. I want to tell you now two brief stories really that will show you what a transformative impact the APDA, and that's really you, all your contributions and your support and APDA leadership support have made to allow us to really work day and night on advancing research for Parkinson's disease, and really to find cures. That's what our center is all about. And this first story actually it's about really a fundamental question in Parkinson's disease. As you all know, one of the key problems in Parkinson's disease is excess production of alpha-synuclein. Alpha-synuclein accumulates in the Lewy bodies in the brain of Parkinson's patients, and it's pretty much found in every Parkinson's patient's brain. And so a key avenue really to new medicines is to be able to reduce the synuclein levels in the brain. And so there are a lot of companies working on antibodies to remove synuclein aggregates, after synuclein has aggregated in the brain, to remove that, to clear it. But we thought actually a more direct and really the best way would be to turn down alpha-synuclein production upfront. And so the question is, how would you dial down synuclein production in brain cells? How can this be done? And thanks to funding from the APDA with the COTIAS Award, we were able to find a transcription factor, actually the first transcription factor of the alpha-synuclein gene. Transcription factor is a genetic switch that turns on activity of the gene. And so we found that this GADA transcription factor turns on alpha-synuclein production. And that really gave us the idea to, well, that's great. This is how this normally works, but can we find medicines? Can we look for chemicals that can activate a switch to turn down alpha-synuclein production? And we did a high throughput screen. Actually, we are really committed to advance with urgency as fast as we can. And so we said, let's screen first all medicines, more than 1,000 that are already approved for other diseases, and see if any of them could activate a switch to turn down alpha-synuclein expression. And what we found is actually very surprisingly that a certain type of asthma medicines called the beta-2 adrenoreceptor agonists activates a switch that reduces alpha-synuclein production. It doesn't completely turn it off, but it reduces it somewhere between 30 to 50%, depending on what cell type you're looking at. And so this was super exciting. That made a lot of waves, and we published this in Science. And I'm excited to say that there are now several beta-2 agonists already in phase 2 clinical trials with the Australian Parkinson's mission, as well as with biotech companies. And we continue to work on beta-2 agonists, and we are working on developing a new generation that is particularly powerful and brain penetrant. So it was really the COCHIAS grant that allowed me to get started to work on alpha-synuclein, and I'm really super thankful for that. I also should say, we've been so this was in 2004 that I received the COCHIAS award, when I actually just was starting my laboratory at Harvard. And so that was a very difficult time. It was also a very new type of laboratory where we, from the beginning, we were really the only laboratory that was combining molecular genetics and computation, bioinformatics. And so it was really with the COCHIAS award and additional federal funding that we were able to get started. So thank you for making this possible. And very briefly, there would be many stories to tell, but time is short. I'm just going to tell you one other thing where APDA really has greatly helped us to attack what I think is really another absolutely fundamental question for the disease. And that is, we know a lot about the genetics that predispose healthy people to develop Parkinson's disease. But I'm a physician scientist, and the people who are coming to my clinic, they already have Parkinson's disease, right? And so in a way, it's too late to prevent and to think about what made them develop Parkinson's disease. What we really want to have is medicines that stop or slow the progression of the disease in people who already have the disease. And so we decided that actually we need to really change the way genetics is done, and we need to go from susceptibility genetics that look for risk factors to actually look at genetics that tell us what is driving the disease in patients. And it's really the disease course over time that's the main driver of a patient's well-being, and that's also most important for clinical trials. And so in the schematics, so with this new type of progression genetics, we want to look at the patient genome and then, based on the genetic glitches, see what glitches cause a patient to progress more rapidly than others. And some Parkinson's patients progress really fast, some really slow. We discussed this. And if we could develop a medicine that turns all fast progressors into slow progressors, that would be absolutely incredibly helpful, particularly if we could keep many patients in the motor stage of PD. We could do so much more with our available treatments for quality of life. And so we set out to do this. We did a genome-wide screen. So far, we found 6 loci, 6 progression genes that are linked to a rapid progression of Parkinson's disease. And these are now prime targets for medicines designed to counter them, to slow down disease progression. So we're working very hard to finding all progression genes and then medicines that correct some of these progression genes. And this has been a long journey working with APDA, so there's much to tell. I just want to finish with 2 things. I'm actually really proud and and excited that I had the fortune to found 2 APDA centers. First, the Harvard and Brigham and Women's Hospital APDA Center in 2018, and I was directing this until a year ago. Then we moved to Yale, and I'm excited to say that APDA recognized Yale for the first time with an APDA Center for Advanced Research. And so we're now all settled in and firing on all cylinders. And so we'll keep working hard in the future using genomics and AI and machine learning to develop drug targets and precision therapeutics. And I want to thank you for tuning in, and thank the people in my lab and my center for doing all this work. And if you're ever at Yale, come and visit. It's a beautiful place. Thank you.
Wow, thank you so much. APDA is forever grateful to Dr. Scherzer for all the work that he's done for Parkinson's research, and for utilizing APDA funding in such a robust and fantastic way. So looking forward to hearing Dr. Scherzer come back for the Q&A. And until then, we're going to introduce our next speaker, who is Dr. Giulietta Riboldi. Dr. Riboldi is Executive Director of the Marlena and Paolo Fresco Institute at NYU Langone Health in New York City, affiliated faculty in the Department of Genetics and Genomics at NYU, and the Director of Clinical Research and Genetics Movement Disorders Division at the Fresco Institute. She completed her MD and PhD, as well as her residency in neurology at the University of Milan in Italy, and a research fellowship at the Motor Neuron Center at Columbia University in New York. Her current research leverages genetics and genomics to understand the pathogenic mechanisms of Parkinson's disease and other genetic movement disorders. Dr. Riboldi was the recipient of an APDA postdoctoral fellowship in 2018 for her work on the role of GBA mutations in the pathogenesis of Parkinson's disease. And this got her on her way to study the discovery of new genetic variants associated with Parkinson's. Thank you so much, Dr. Riboldi, for being here, and take it away.
Thank you so much, Dr. Gilbert, and thank you so much again for the invitation to be here and discuss about the contribution that the postdoctoral fellowship from APDA really had in the development in my research program, my research career. Starting with a little background, as was just said, my training started a little bit far from here, across the oceans, where I completed my medical school and my residency in neurology at the University of Milan. And I was lucky enough to be involved early on in training in genetics of neurodegenerative diseases. In order to pursue this interest and passion that I have for research in this field, I then crossed the oceans and I completed a postdoctoral research fellowship at Columbia University at the Motor Neuron Center, where I was trained more in the molecular mechanisms of neurodegenerative diseases. But it was not until I landed to NYU that I was able to invest all this knowledge in the field that I was really more passionate about. It was the one of Parkinson's disease and movement disorders. That I was just said here at NYU, I completed my fellowship in movement disorder, and then I stayed on as faculty, and I'm now an assistant professor, and I'm serving as the director of the clinical research and genetics in movement disorders and the executive director of the Fresco Institute. However, looking back, the main cornerstone for the beginning of my research program has really been the postdoctoral fellowship that I received in 2018 from APDA. That really start off my research involvement, and also allowed me to start working on the field that I was more interested and passionate about. It was the role of genetics in Parkinson's disease. In particular, I start focusing on the study of one of the most now important genetic risk factor for Parkinson's disease, which is the gene GBA. This is an interesting gene that encodes for an enzyme, a protein in the cell that is important for the metabolism in the cells. And interesting enough, this gene has been long known to be associated actually with another condition that is called Gaucher disease. This is a rare disease that has apparently nothing to do with Parkinson's disease. It's usually presents in kids with symptoms like enlargement of the liver, of the spleen, involvement of the bones, of bleedings, and it's also characterized by the accumulation of dysregulated immune cells called macrophages in specific targeted tissues. Interesting enough, about 20 years ago, clinicians start noticing that in people with Gaucher disease and their family members that could therefore be carrier of the same mutation, there seems to be an increased frequency of Parkinson's disease. There was a little bit more than what I was expected. And so they started this relationship, and that was proven to be true. So much so that today we know that GBA is one of the main genetic risk factors for Parkinson's disease. However, there were many questions that were needed to be answered around these findings. And first and foremost, about the connection between the mechanisms that is causing Parkinson's disease and the presence of these genetic mutations. But also because of the role of these genes in the immune cells, we were wondering whether there was a dysregulation of the immune system also in patients with Parkinson's disease and GBA mutations. And finally, we know that this gene is a risk factor for the disease, meaning that it can increase the risk for a person to develop Parkinson's disease, but not everyone who has the mutation in this gene will eventually develop Parkinson's disease. So one of the main needs in this field is really to identify biomarkers that would be able to detect or predict who among the people who have the mutation in GBA mutation will eventually develop Parkinson's disease. So with the grant that I received from APDA, I really tried to explore, especially the last two of these questions. And so we enroll a cohort of patients with Parkinson's disease with and without GBA mutation, and patients without Parkinson's disease with and without GBA mutations. And we start looking first at different biomarkers that could help identify and detect patients that would go on and develop the disease in the presence of the mutations. And the first things that we looked at were differences in terms of the eye movement in collaboration with Dr. Acker and Todd Hudson at our center. Why? Because we know that in Gaucher disease, there are specific abnormalities of the eye movement. And so we wanted to see if those could be detected also in patients with Parkinson's disease and the mutation. And interestingly enough, we were able to find that in patients with Parkinson's disease and GBA mutation, there were specific changes in the frequency and in the amplitude in the horizontal and vertical eye movement compared to patients without the disease, but with the GBA mutation, and also compared to people with Parkinson's disease without GBA mutation, meaning that they are very specific for PD associated with GBA mutations. Now we are studying the markers in larger cohort to see if those can be utilized as early biomarker for phenoconversion. We then extended our analysis to a larger set of non-motor features associated with Parkinson's disease, like REM sleep behavior disorder, dysautonomia, loss of sense of smell. And we were able to build, leveraging our cohort and a larger cohort available from a public available database, we were able to build a model to predict the presence of GBA mutation in patients with Parkinson's disease using the clinical data. And this allowed us also to identify specific demographic and clinical features that may be very much relevant to identify patients with this specific genetic mutation, and maybe to predict also the presence of mutations or the development of disease in patients with the GBA mutation. In parallel, as I said, we were interested in looking whether in patients with Parkinson's disease and GBA mutation, there could be a dysregulation of the immune system and how this could inform in terms of possible mechanism and pathways that were involved in this condition and in the pathogenic mechanism of Parkinson's disease. So in order to study that, we collected blood samples from the cohort of patients and controls that we enroll in our studies, and we look at differences in gene expression in both the whole blood and also from isolated specific immune cells called monocytes from these blood samples. And so as we characterize the gene expression that can point out to specific pathways, specific changes in the function of the cells, we saw that in patients with GBA mutation and Parkinson's disease, compared to people with GBA mutations without the disease, there were this regulation of genes that were pointed out to an impaired function of specific function in the cells, including a specific activity of the immune response, a specific processing of the secretory granules that are contained into these immune cells, as well as a process related to metabolism of RNA, meaning of the genetic informations. And interestingly enough, we were able to see some of these changes also when we look microscopically at the microscopic levels in these cells, suggesting that there was a dysregulation and impairment in the immune cells in patients with Parkinson's disease and GBA mutation. These, of course, open up even more questions that we gone on and investigated through following up research studies and programs that were supported by different agencies. In particular, we wanted to try to understand what was determining these changes in the expression of genes in the immune cells. And so we are studying now the contribution of epigenetics, which is a system that controls the expression of the gene at the level of the DNA, and that can be influenced by the interaction with environmental factors such as specific lifestyle or exposure to smoke or environmental toxins. And by the interaction with the environment, epigenetics can change the gene expression. And so now we are looking at the interaction between epigenetic changes, exposure to certain factors, and the changes in the gene expression in the immune cells in a large cohort of patients with Parkinson's disease, and also patients with prodromal features of Parkinson's disease, meaning features that can predispose to the development of the disease, in order to try to see how these three factors interact with each other and can contribute to the development of the disorder. And secondly, since we saw that there were these significant changes in specific genetic forms of Parkinson's disease, meaning the one related to GBA, we then ask whether specific changes or other different changes can be found also in other genetic forms of Parkinson's disease. So I just started studying a very large group of patients with young onset Parkinson's disease. This indicates a disease that starts before the age of 50 years. And we know that when the disease starts early on, it's more frequent to be able to identify specific genetic mutation. So we are now studying patients with young onset Parkinson's disease and try to characterize their genetic profiles and correlate that also with the different gene expression and changes in the immune system correlated also with biomarkers to try to detect the mechanism that is causing specifically young onset Parkinson's disease and identify different subtypes of the disease going in the direction of a precision medicine, as was stated also before. So to summarize, really the postdoctoral fellowship that I received from the APDA has been the cornerstone for the development of our current research program that is now leveraging national and international collaboration and is building up a very large biorepository at our centers, through which we are conducting different research and programs to explore and to try to contribute in the understanding of the role of genetics in Parkinson's disease, but also through the interaction of other mechanisms that can work together, such as the response of the immune system, as well as the interaction with the environment. So with that, I would really like to thank first and foremost APDA for the great opportunity that I received with that postdoctoral fellowship grant that really put the foundations for for my following development in research and the development of my research program. And also would like to thank all the people in my research group that really day in and day out make all this work possible. Thank you.
Thank you so much. That was amazing. Thank you, Giulietta, for all your work and for collaborating with APDA on these fascinating projects. Thank you so much. So finally, we have our third APDA-funded researcher, Dr. Sunil Kumar. He is assistant professor in the departments of chemistry and biochemistry and at the Nobel Institute for Healthy Aging at the University of Denver in Colorado. He received his PhD at Clemson University in South Carolina and then did postdoctoral fellowships at Yale and NYU, the two universities that Julieta and Clemens are at. The central theme of his lab is the design of tools, synthetic, biophysical and in vivo, to gain mechanistic and therapeutic insights into various diseases, including Parkinson's disease. Dr. Kumar received an APDA research grant in 2021 and then again in 2022. And these grants turned out to be seed funding for his work on identifying drug-like ligands that could specifically target and disrupt the aggregation of alpha-synuclein, which is a process that is directly associated with the development of PD. So Dr. Kumar, here you are, and take it away. Thanks so much.
Thanks, Rebecca, and thanks for the invite. And it's been great. I learned a lot even from last two presentations, and I'll just talk about the work we have been doing in our lab. I'm still in the early stage of my career. I'm still an assistant professor. We started our lab in 2019, just before the pandemic, and I'm really appreciative of the grant we got. That was my first grant as well from anywhere, from APDA, and that really helped us in starting my career in our lab in order to make a lot of stride in the research, which I'll just talk about now. So just to make a disclosure as well, so I'm also we have a startup company based on the work we did in our lab. We patented it, and now I'm partner in that. I have equity in. It's called Neurofold Therapeutics. So just to give you a little background, it's been talked alpha-synuclein earlier as well. It's a protein which is a biomolecule, if you will. And you can see in this picture, it's very, very important in regulating the release of a neurotransmitter, which is a dopamine. So it's important that it releases it. It helps in its release so that the neurons, they can connect with each other, talk with each other in order to regulate motor functions. But when this protein start aggregating, means it start clumping in our brain, the process of release of dopamine significantly decreases. And that could cause the early onset of Parkinson's disease, and then moving on to the Lewy body dementia. So, and my training was in organic chemistry, and as a chemistry person, I used to see that process, which you are seeing on the right side, as the clumping of these biomolecules together, and then they keep on clumping and form these at the end amyloid fibers, which are, I give a analogy of plaques. They get deposited in people with Alzheimer or Parkinson's. Now they are formed because of these clumping of these proteins. Now, what we thought is, can we mimic these proteins, the clumping, with an artificial protein? We can make them, design them in our lab. And can we do that? We just need to somehow mimic them. And that's the idea we came up with. And if you do that, we could stop this process of the inhibition of this aggregation. And you can see we published heavily in a number of very decent papers. And these are the molecules. One example, which you are seeing down here, a chemical structure of a artificial protein, which you are seeing on the right, and that mimic the protein which is clumping on the left. So they are mimic of those proteins. So after that, we did a lot. Our lab is has a lot of models of Parkinson's disease, going from very small organisms to all the way to mouse models, as you can see here. So we systematically, using these molecules, we test them to cure Parkinson's disease in mammalian cells. Then we go to primary culture neurons. We have a lot of C. elegans models, and then mice model. So all these work we have done in our lab, and we did it in a very systematic way in order to make sure that we take out any problem with these ligands in terms of their toxicity, in terms of other hurdles. We systematically take them out to move them forward towards a clinical trial. And as I said, Neurofold Therapeutics is our another startup where right now what you are seeing on this graph is the pipeline, where we are very close to the preclinical. We have these lead molecules which are very close to the preclinical stage. and hopefully in the next year or two, we will moving towards the phase 1 clinical trial. And just to give an idea, at the beginning, this is my lab in 2019, and as I said, APDA, that was my first grant 2021 in summer. I remember the day. I was very happy we got our first grant, and we only had 6 people in our lab at that time. But now, because of that grant, that was a catalyst. We have gotten almost $5 million in funding till now. And our lab, as you can see now, there were like 6-7 people to there are almost 20 people. And we are working day and night in order to make sure that we can push these molecules which developed in our lab towards the clinical trial to help Parkinson's patients. And I really appreciate, as I said, it's very close to my heart, APDA, because that was the first grant I got. And from that, everything started turning for us in order to move forward these molecules. So thank you very much.
Thank you so much. That was amazing. Thank you to Dr. Kumar, and thank you to all our presenters who really painted wonderful pictures for how PD research is moving forward. So thank you so much. And everyone's back to participate in our Q&A. So thank you so much again, and we will try to answer as many questions as we can. So the first question actually is really related to Dr. Scherzer's talk. Glenn asks, is it believed that all Parkinson's cases, so whatever the cause, that there is a genetic component to it, environmental, both, is there a genetic component to the rate of progression? Is that what you were trying to establish? Yeah.
So I think there is good evidence that the really fast progressors have a genetic component, and that also the really slow progressors have a genetic component. For those patients sort of in the middle of the road, I think it's less clear. I'm not sure about that. But it's actually really interesting actually to, I talked about, and Julieta and I talked about genes that GBA, for example, make patients progress more rapidly. But interestingly, LRRK2, which is the second most common Parkinson's gene, actually is all the data we see and we've seen suggest that those patients actually have a really slow progression. So it's a very interesting area, and I think many more targets and hopefully drugs will come out of it.
Great, fantastic. So we have a question that was actually submitted during registration, which I'm going to ask Julieta to answer, which is, this is a person who has has 2 brothers with Parkinson's, and feels most likely has some kind of genetic situation in her family related to Parkinson's. What would your advice be to somebody like that in terms of their own risk, getting involved in research? How would you address this sister with 2 brothers with Parkinson's?
Absolutely, yes. This is questions that we encounter many, many times, both in the clinical practice and also in the research setting. So definitely, it's a very important question. And definitely, for sure, if there is a strong family history, this is very suggestive that the possibility of identifying specific genetic mutations. Having said so, we know that we can find genetic mutation even in people that do not have a family history, but definitely when there is a strong family history, like in this case, could be very much the case. Now, for people who do not have family symptoms of Parkinson's disease, the indication of getting a genetic testing needs a very thorough discussions, I think, with experts in the field. So because we have always to consider the pros and cons in this context. One thing to consider is that for now, we don't have specific treatments that could target specific genetic mutations and that would prevent the disease. We are trying to go in that direction with research, but we are not there yet. And also the other important thing to consider is that the 2 most common genes, GBA and LRRK2, that was just mentioned, they are risk factor, which means that not everyone who has a genetic mutation eventually will develop the disease. So I always discuss that with people that ask these questions, because it means that someone can get tested and maybe be positive for the gene, but never develop Parkinson's disease in the future. So I think that this is a bit considered, especially because we don't have preventive treatment right now. So having said so, I think it's extremely important to be involved with research, so that to contribute to get there to preventive treatment, and also to be kind of in the forefront in case of new treatment becomes available. In terms of getting tested, I think that the pros and cons have always to be weighted, also in consideration of how would be a personal response in knowing whether you have a genetic mutation that can increase your risk but not necessarily determine a disease.
Yeah, it's, I think, much more complicated than people realize, and really does require an involved conversation about any particular person's situation. Thank you so much for that. Dr. Kumar, so here's a good question for you. You discussed sort of taking a research approach from the start to a potential clinical trial. And we've had a number of questions that want to further understand what the time frame of that would be. People get frustrated by how slow research goes. Can you give us some indication about timings from start to maybe a drug being available? How long would that potentially take?
That's a great question, Rebecca. And now I'm more involved with the patients when we talk to them, and it's obviously very frustrating that there is still no drug yet, and there is a lot of effort, but the outcome is not there yet. It doesn't mean that it won't come, but from our perspective, it comes down to a couple of things. One is from our research and our company, we started a startup. We are now trying to get as much money as possible in order to move it forward, because it's directly related with that. One thing, how much money we can raise in order to start those trials. But if everything goes all right, as our company has seed money now, a lot of it, and my lab also has a lot of grant funding. So we are moving swiftly right now without any problem. For next, everything goes fine and I'm telling from our perspective, and I can give an idea about how others are doing, but it will be in next 2 years, we will start the clinical, even quicker than that, 1.5 years, the clinical trial for our drug, and then it would take another road, like it could be 3-4 more years before we see the drug, if everything goes great. And one big hurdle, as I am pointing out, it's the finances, because as you can see, it requires a lot of financial commitment in order to move them forward. And even you try a lot harder in order to get it, there is no guarantee, both from the company and the grants, that it comes in a timely manner. So that's one big challenge always. But there are other companies and drugs which are a little bit ahead than us. There are at least in clinical phase trial 2, or almost finishing them, and we know them because we are connected with them as well. The people in our company, they are consulting for them, and right now we have pretty good knowledge that they are almost finishing actually clinical phase 2 trials. So that will be just one clinical phase 3, and hopefully that would be much I guess quicker, maybe in a year or 2.
Thank you very, very much. Research is slow, and we'd love it to be faster. Thank you so much, Dr. Kumar, for that answer. Along those lines, I'm going to ask Dr. Scherzer to answer the next question. There's a lot of talk these days about federal funding of research, and if you would address sort of in generalities, what are your thoughts about funding? I should say APDA does not receive federal funding, so our funds are philanthropic, and we haven't had any cuts that we are worried about. Federal, however, is different. And Dr. Scherzer, if you could just sort of talk in generalities about the importance of funding to your center and to research in general.
Yeah, it's really a sort of an all team effort. This making progress, requires great ideas, immense commitment, and it requires funding. In fact, for our research, it's when people ask me what any barriers you have or anything as obstacles, it's really we know what to do. We know what we want to do and how to get to new targets, how to get to new therapies. And it's simply a question of scaling up the assays, the methods we have in parallel, do it faster and more rapidly. And so it's important. Federal funding is crucial. We have been very fortunate to, over the more than 20 years, get very substantial federal funding from NIH and the Department of Defense. And I'm sure that will continue. There are always twists and turns. There are always ups and downs, but it's really, that's really part of science. You encounter challenges and you need to raise to them. Philanthropy and foundations are a critical partner in that. Typically, NIH is really critical for very large funding and sort of typically geared towards more conservative funding that sort of makes an incremental progress. But what we really want to do is sort of make quantum leaps, big jumps forward. And this kind of risky science needs foundations and it needs philanthropy. So it's critical that we all work together. Our audience, we need you. Foundation, we need you, and we certainly need the NIH and the DoD to keep funding Parkinson's. Parkinson's, of course, is the most rapidly growing brain disease, as you know, and actually the rate of growth has outpaced now Alzheimer's disease, and this is really going to lead to an immense burden, really a pandemic around the world for the U.S., but also for other countries. For example, I've been thinking a lot about India recently, and because the population is aging and living longer, actually there are going to be 20 million Parkinson's patients expected in India over the next decades. And so it's really critical for everybody in the world. And, whatever party you vote for, whatever you're interested in, everybody needs to be healthy. Everybody needs a medicine. And so we just need to work together as fast and as best as we can.
Thank you so much for that. So Julieta, we have a question for you that came in through registration, which is, could DNA repeat expansions be a causative factor in the development of Parkinson's? Interesting question.
Very interesting question, very sophisticated question, I would say also. So as far as we know, DNA repeat expansion are now being identified as the cause of many also neurological diseases. Not so much so in terms of Parkinson's. They're mostly found in other conditions like Huntington disease, some form of spinocerebellar ataxias, some forms of progressive myoclonic epilepsy, but in pure Parkinson's disease, they are not one of the main cause. There are usually other genetic changes that we see in genes that have been associated with Parkinson's disease. Having said so, some of these conditions, considering, for example, some form of spinocerebellar ataxia, but also some presentation of Huntington disease, sometimes they can present initially with symptoms that can resemble the ones of Parkinson's disease or Parkinsonism. So there could be something that when we do genetic testing, when there is a suspicion of a genetic cause, should be taken into consideration as a possible cause when other genes that have been tested are negative.
Fantastic. So here's a question from Keith directed to Dr. Kumar. So if you could summarize your talk a little bit just to highlight the highlights for everyone. Keith asks, have you discovered a synthetic protein that reverses alpha-synuclein clumping? And
yes,
tell
that's
us
a
a
great.
little bit about that. Yeah, go ahead.
That's a fantastic question, and the answer is yes. But I'll just tell you two things. One is we are not the only one who's doing that. There are antibodies, immunotherapy. They have done it, and the effect is okay. It's not great. There is effect. But what we did is another extra thing is we found these synthetic proteins which we have in our lab. We can target the middle stage, which we call the oligomers, which are very, very toxic and much more worst effect on the neuron degeneration and everything in comparison to just dissolving declumping the fibers. So that was our approach, and we saw very, very exciting complete effect in the mice models so far. And so the answer is we are working at a two-way approach instead of just dissolving the clumps. And the answer is yes, we have that molecules. So the other big thing is with our molecules, these synthetic artificial protein we call, they are much smaller than the antibodies. So if you give the weight, for example, antibodies, they are 20 to 30 fold higher molecular weight than our molecules. And when it comes to that big molecular weight, they cannot cross the blood-brain barrier, which is required, a key thing to reach to the toxic target in the brain. And our molecules, they are much easy to cross that the barrier which is in the brain. So all these things, we compared it with the immunotherapy, and it's very exciting to see that they are outweighing that.
Great, fantastic, very exciting. So we have a general question from Dale, and I think any of you could answer this, which is it fair to say that your research efforts are moving away from the strategy of replacing dopamine as the primary approach to Parkinson's? So maybe, Clemens, you could give your answer to that.
Yeah, that's right on point. We're fortunate to have actually almost 40 medicines. Most of them, in one way or another, replenish dopamine in the brain, but they don't slow down disease progression. And so really all the research you've heard about today is directed at disease-modifying therapeutics that address one of the underlying causes of the disease, one of the genes driving the disease, for example, like alpha-synuclein or GBA. And yeah, so there's a lot going on, and a lot in the pipeline, as you've heard from Sunil and our center and others. And so I'm really optimistic that some of these new compounds will succeed in clinical trials and really change the way we treat Parkinson's.
Fantastic. Anyone else want to comment on how their research may be sort of moving from a symptomatic approach to treating Parkinson's, getting rid of some of the symptoms, to a neuroprotective approach where we're really getting to the bottom of what is causing Parkinson's and addressing that?
I can say 2 words. Yeah, no, absolutely. As it was just said, the medication, the cholinergic medication are symptomatic, and we are really trying to find something that could address the mechanism and so slow down, or in best case scenario, even stop the disease progressions. So in that direction, also, that's why a lot of the research is also try to determine and define more and more the different types of Parkinson's disease, because we know that Parkinson's disease, even from the molecular perspective, is not necessarily all the same for all the patients. And so understanding the contribution of genetics, of exposure to environmental factors or other mechanisms like inflammations, and how this plays out in different proportion in different patients can be also very important in that direction, because we may not have just one treatment then they slow down Parkinson's for all the patients, but we can have probably different treatment that slow down Parkinson's disease for different patients depending on what is the mechanism that is causing the disease in their case.
Fantastic. Thank you so much. We are amazingly at the end of our hour, and so we are going to have to wrap up now. I wanted to thank you so much, all three of you, for fantastic information and for really getting us to think about how Parkinson's disease may yet be cured in the near future. So thank you so much for all your efforts in that direction. If you know someone who missed today's program, if you joined late, if you'd like to watch again, this recording will be available on our YouTube channel. And don't forget to subscribe when you're there, because that is how we know what you like. For additional information and resources, please visit our website at apdaparkinson.org. I wanted to inform you that our next Dr. Gilbert Host will take place on Wednesday, May 28th. We're skipping April. May 28th at 2 p.m. Eastern Standard Time. We're going to be joined by our special guest movement disorders physician, Dr. Julieta, and she will be talking about Parkinson's disease impact on other medical problems. So if somebody has a medical problem such as heart disease, diabetes, or cancer, how does that interrelate with them having Parkinson's disease as well? So it's going to be a fascinating discussion, and be sure to register and be available for that as well. And be sure to stick around now for a few final messages. Before you go, I want to say thank you again to all our guests. Thank you for joining us today and for all your questions. We hope to see you soon on another APDA program. Have a great afternoon.
Hi, I'm Leslie Chambers, President and CEO of the American Parkinson's Disease Association. Thank you so much for joining us today on our program. Every day, APDA is committed to providing helpful programs just like this one, as well as a wide variety of support groups, exercise classes, and educational programs to support the Parkinson's disease community. Whether online or in person, APDA is here for you every step of the way. You can find all of our upcoming virtual events on our website at apdaparkinson.org/events. We keep you posted on our social media channels as well, so make sure you follow us there. You can also connect to local resources and events through our nationwide network of chapters and information and referral centers. Remember, we're just a click or a phone call away. If you've enjoyed today's broadcast, I hope you will consider making a donation to help keep programs like this possible. Every gift, large or small, helps APDA support people living with PD through thoughtful programming, reliable resources, groundbreaking research, and more. Please visit apdaparkinson.org/donate today to support our critical work for those with Parkinson's. We are so grateful for your generosity. Thank you.